COVID-19 has been on people's mouths for some time, from a TikTok compilation to a disturbing footage filmed by news outlets depicting thousands suffering. The virus has taken the world by storm. However, in order to defeat our enemy, we must first know it. So, how does the coronavirus take over our cells? And what is the chemistry behind it? The coronavirus consists of four main categories. These four categories are the envelope protein, the envelope, the nucleocapsid, and the viral RNA. These four components are essential for a virus and is shared among various species of viruses. Starting with the outside layer. The outside layer consists of the envelope and the envelope protein. The envelope is a protein layer that coats the virus and protects the component inside while the virus is traveling either inside or outside the body. The envelope protein, or more commonly known as the spike protein, is a spike per se that is used to infiltrate the cell when it reaches it. In the inside, the two main components are the nucleocapsid and the viral RNA. The nucleocapsid serves as a secondary protection for the genetic material of the virus while it is being transported. The viral RNA contains the instructions in order to replicate the virus when it reaches a cell's nucleus. Now, what does this have to do with chemistry? In order to discuss that, we now need to move to the process of protein synthesis. Protein synthesis occurs in a two-step process: transcription and translation. Transcription is the process of converting a DNA to an RNA. As we all know, a DNA contains genes, or a section of the DNA that is responsible for coding for a specific protein. However, a gene stored in a DNA cannot be used by the cell. Therefore, it has to be converted into an RNA. This process is known as transcription. In translation, the RNA produced through transcription is interpreted and is made into an amino acid chain. Protein is made through a condensation polymerization reaction between amino acids. However, since there are so many, how does our body know which to combine in order to produce a protein? The answer lies in the RNA. The RNA is composed of nucleotides, and 3 nucleotides make up a codon, or a section that codes for amino acid. Inside the ribosome, or the organelle responsible for coding proteins, each codon is coded for amino acid, and an amino acid will be attached to the chain as the codons are read. If the entire codons of the RNA is read, then the ribosome releases a full chain of amino acids, which can be converted to a protein later on. Now let us examine how these amino acids combine in the process of condensation polymerization. In order to explain how amino acid chains are made inside a ribosome, I will take two hypothetical amino acids, alanine and glycine. Every amino acid existent shares two functional groups, the COOH group and the NH2 group. Let us move to the workbench to see how these two groups bond together and form a chain. Now, when these two amino acids bond together, the OH group from one amino acid bonds together with the H group from another amino acid. These two groups combine and form a single water molecule. Meanwhile, the vacancy of the OH and the H molecules allow one vacant space for bonding together. Therefore, it allows the C, which is not visible here, the C and the nitrogen to bond together. With this process, the end product of the amino acid chain looks something like this. The bond made through this reaction is called an amide bond. As this reaction progresses, more and more amino acids follow the same reaction and add to the chain. And as the chain progresses, each amino acid is added through the instructions in the RNA. Therefore, through this reaction, a protein is made. Now, recalling that viruses are made out of proteins, let us now examine on how viruses hijack this system and use it to replicate themselves and invade our body. In order to discuss how a virus invades our body, let us first discuss how a virus invades our cell. In the outside, the virus has spike proteins. These spike proteins contain receptors that bind with our receptors of our own. When these bind together, the virus is able to enter our cell. Inside the cell, the virus is now free to release its RNA. As stated earlier, the RNA contains code to produce a specific protein. A viral RNA is coded to produce viral proteins. And when this RNA enters a cell's nucleus, it is able to hijack the system and use its own RNA instead of the body's to use our body's functions to replicate itself. As mentioned before, RNAs dictate which proteins are produced through their instructions. When a viral RNA hijacks our body's production system, then a new chain of amino acids are added instead of the original body's programming. Therefore, the body does not produce enzymes or proteins beneficial to our body, but instead replicates the virus. And as the viral proteins are made, they are then reassembled in the Golgi apparatus. This is one cycle of a virus being replicated. This repeats for thousands and thousands of times until the cell cannot hold enough viruses. Then the cell will die and the virus moves past on, searching for another target.